Hello everyone, welcome to Focus Weekly of Rouse IAS. Today on 9th of January 2025, I'll be discussing the recent updates in the subject of science and technology. In this particular time frame, I have shortlisted 6 topics for us, and these topics are going to be dealt in detail as per the UPSC standards, and we'll also be looking at some possible questions from these areas that can be asked in the prelims as well as mains examination. The topics that I'm going to discuss are Google Willow quantum computing, and I'll also going to be discussing the sonobuoys initiative, where India and USA are going to co-develop and co-manufacture new kinds of undersea surveillance devices. MicroRNA, this was also popular from the context of Nobel Prizes 2024, but it is still very relevant in the month of January. There have been news related to this in the past 1 week. Twigstats is an interesting genetic analysis technology. This is where we try to merge computer science and genetic analysis and how it can be helpful. What are the possible applications? We'll see all of them. Then we'll talk about hydrogel targeted therapy developed by IIT Guwahati. It's an interesting therapy developed for breast cancer treatment, and it is considered to be a better innovation compared to the incumbent technologies. Then lastly, we'll talk about PSLV C60. This is the latest mission of ISRO, and what are its objective, what landmark achievements it is expected to make, and what are going to be its contributions in overall space commerce, overall space development of India. We'll discuss all of these points here. Now, starting from the first point that's related to Google Willow quantum processors, let's start with this discussion first. But before I actually start with what is Google Willow, I have to first give you a brief comparison between traditional computing and quantum computing. What really makes traditional computing, inferior to quantum computing, and what really makes quantum computing so important and so relevant from the future point of view. So guys, if I just ask you a question, if I just ask you a simple question, let's imagine if you have got a maze in front of you. You've got a maze, you have one starting point of the maze, one end point, and you have a lot of bars, a lot of lines across the screen, and you are supposed to travel, find a possible path, start from the start position to the end position. Now, how normally one would try to do? They'll start from the starting position, and you'll start scribbling through the maze and try to find out what possible route. So you will find the route 1, and you find an obstacle, you stop, you eliminate that route. Then you start with the second route, you try to move to the route 2, you will find another obstacle, then you stop, you eliminate that route. Then you start with the third one, and so on. You may think a little smart where you start with the end position and try to find the route, but again you may find some obstacles, and that's how you'll keep eliminating different routes, right? So likewise, you may start, and with every attempt you'll have a hurdle, and the moment when you hit a hurdle, you'll stop and you start again. This is what normally or traditionally we would do. But imagine if we create a system where the moment you look at the maze, you look at the starting position, look at the end position, and all the possible routes. Let's suppose there are 100 possible routes, and only one possible route is the correct one. So you create 100 possible routes, and only one route is there which is going to take you to the end position. So traditionally we'll go one by one, but what if we create a system where we can find or trace all possible routes and then pick up the best route that takes us to the end position? This is the fundamental difference between a quantum computer and a traditional computer. In case of a traditional computer, every information that you take, every information that you provide, every information that you receive has to be processed in a sequence, one by one, one after the other. So let's say, for example, if you ask a traditional computer to actually perform a calculation which involves calculations from digit 1 to a very high number in billions, a traditional computer ideally will start from the first digit, will see how many zeros, how many numbers are there, and will take up all the steps one by one. Will see the digit positions, 10th position, 100th position, 1000th position, right, millionth position. Likewise, it will observe, and then it will come up with a solution. Whereas if you compare a quantum computer in this, a quantum computer will analyze all the positions in a single go and give you the solution by looking at all the possible solution, and will pick up the right solution and present it to you. So if I would try to give you a very simple way to understand what makes quantum computing better, is that a quantum computer has a ready-made solution to a problem all the time. It is just that it has to pick up that ready-made solution and then provide it to you. Whereas in case of a traditional computer, once a problem is given, a problem has to be sorted from one step after the other. It may take, minutes to solve that problem. It may take days. It may even take years to solve a problem, depending on how complicated the problem is. So suppose if I ask a quantum computer, or if I ask both traditional and a quantum computer about the total number of stars in the galaxy Milky Way, a traditional computer would ideally start with the first star and then start coming up with an actual number. Whereas a quantum computer will have all the numbers already created. It will just find out the most acceptable or the most tentative number and give it to you right away. So basically the thing is, in case of quantum computers, or in case of, first if I talk about traditional computers, in case of traditional computers, they follow a sequential, or you can say a systematic algorithms to solve a problem. For any information, they have to go one step after the other. But in the case of quantum processors, they happens to be following the principle of superposition. Now, don't ask what is superposition right now, but we can have a separate class. You can write in the chat and let me know if you want a separate understanding of superposition principle. It is a topic related to quantum computing, quantum mechanics. Just consider that at a very small atomic stage, at the stage of an atom, or at a stage even smaller than an atom, at the stage of electron, proton, neutrons, particles behave in a very different way how they behave normally here, right? So when you talk about the laws of physics working around us, it is very easy to comprehend, it's very easy to understand, very easy to observe. But when you apply the same laws of physics or any physical phenomena at the very small atomic scale, we realize that atoms, or these particles inside atoms, they tend to behave very differently, right? So one such different behavior of atoms is called superposition, and because of this superposition principle, the quantum processors do not have to follow a sequence of solution. They don't need to have a sequence of solution, rather they have got a string of solution. And from that string, you need to simply pick up the right answer, pick up the right information, pick up the right solution you're looking for. So when it comes to a quantum processor, because it follows a mechanism of a string of solutions, it follows a mechanism of string of possible outcomes, and you simply have to pick up the right outcome. The outcome is already there, you simply have to pick it up. That way, it helps you to provide any information much, much faster. Guys, when I simply verbally try to explain these things, you may be able to realize that, okay, if a quantum processor is solving a problem, it may be able to solve a problem faster than a traditional computing by 10 times, 100 times. But you'll be surprised to know that when Google introduced their Willow quantum processor, it has been in news since January 2024, but now Google has been able to finalize it. So with Google Willow, they have been able to achieve a level of accuracy, a level of efficiency, where Google developed an algorithm. This algorithm did not have any real life application. It was only developed just to check whether this particular computer or whether this particular processor is reliable or not. So they created an algorithm, and they found out that this algorithm was resolved or calculated by Google Willow in a matter of 5 minutes. So the algorithm that took Google Willow 5 minutes to finish, or to 5 minutes to complete, 5 minutes to solve, a traditional supercomputer. Supercomputers are the fastest traditional computers in the world right now. A traditional supercomputer would be able to do that in what they have mentioned is 10 septillion years. What is 10 septillion years? It is 10 to the power 23 years. These many years it will take a traditional supercomputer to solve the same algorithm. So when you talk about the Google Willow quantum processors, they have been found to be many times, I can't even tell you how many number of times, it is multifold times faster than a traditional computer. And guys, considering the future of electronics, where we are looking for high speed devices, more efficient, more accurate devices, quantum processors are considered to be the most popular innovations of the current time, because they are likely to be efficient, processors that are going to be replacing the traditional processors for in possibly all industries, all kinds of innovations. So this is what Google Willow has been able to achieve, and that is the reason why Google Willow has been there in the news regarding its possible applications, regarding its possible achievements, right? So first is its very high speed. So the first achievement that Google Willow has made is extremely high speed. The speed of Google Willow is extremely high, right? It is very high. Then guys, the second achievement that it has been able to achieve is error correction. Now guys, when it comes to error correction, now error correction is another aspect that is where quantum computing have been found to be better than traditional computing. See, we all understand that when you talk about any electronic devices, it will have some errors. In fact, some of the best clocks, which we often consider as atomic clock, even that also has an error which has to be adjusted in a million years, right? But when it comes to computing, Google Willow has been found to use some innovative error correction techniques, and these innovative error correction techniques have been found to be more reliable and actually have increased and have actually increased its scalability. So with better error correction, it is able to solve algorithms more accurately. It gives you more accurate information. And lastly, there is a term that has got really popular recently. It's called below threshold. There is a term that has got really popular recently. It's called below threshold. What is this below threshold? Below threshold is a popular term related to quantum computing. Below threshold basically means that now the rate at which it creates an error, it is now so less. The rate of creating errors, below threshold, is the rate of creating errors. So like every man-made system creates some kind of errors, the rate at which Google Willow has been creating errors, it is now below threshold. So threshold is basically a certain rate that has been set for quantum and traditional computers. It is for the first time somebody has created a quantum processor which is now within the threshold limit of traditional computing regarding error creations. And with this possibility of creating less errors, it is now possible for us to create real quantum processors by using the system or using the quantum computing technology developed by Google. So these three are the things that makes Google Willow better than traditional computing, right? Now, if I just try to give you some differences between traditional and quantum computers, you can simply write down. I would expect you guys to be writing constantly. Please keep a notebook and a pen handy so that you can make notes. So first point that one should write under this discussion is that quantum computers, they work on superposition principle. In which all combinations of zeros and ones are created at the same time. Right? This is the first point about supercomputers, superposition principle that you should write. So they use the principle of superposition, and by using this particular feature, by using this property of superposition, a quantum processor is having a very high speed compared to any classical processor, right? Now, what makes Google Willow better? So in terms of speed, it is multiple times faster than a traditional computer. In terms of its error correction, it has better ability to correct errors, and its error creation is now below a certain threshold. So it is below threshold error creation that makes Google Willow a better quantum processor of the time. Now, with these possible achievements, what are the possible applications that one can assume for Google Willow? So guys, when you talk about applications, the applications of Google Willow will involve mostly in the area of data, whether it is data management or data analysis. A quantum processor would be very capable of analyzing any kind of data compared to a traditional computer. Secondly, a quantum processor provides much better ability of data protection. Data protection. Like if I compare, suppose if you encode or encrypt any data with a traditional computer will have a certain algorithm that will be, encrypting any information. A quantum processor, on the other hand, using its ability of superposition principle, can actually decode or decrypt any encrypted code of a traditional computer. So in terms of data protection, it is better in decrypting the information of traditional computing, and once a data has been encrypted by a quantum computer, a traditional computer is nowhere going to be close enough to decrypt that data. So data encrypted by a quantum computer is very much secure, is far more secure than the data encrypted by a traditional computer. So data protection, and with these ability of better data analysis, data management and data protection, it has applications in various industries. We can develop better quality drugs for better drug production, drug sequencing. We can use this data for better decision making in the sense of designing. We can design better quality products, we can make things more ergonomic in nature, right? We can also take better decisions in terms of judiciary. We can analyze all the past data of all the judiciary records, and we can study that data and eventually provide all possible solutions or best possible solutions to any kind of cases, any kind of important hearings. Then it is considered to be very safe when it comes to financial models, creating different kinds of financial models, right? Banking sector can be benefited by this. All these are possibilities that will open up through quantum computing, right? So what I think about quantum computing when I talk about its possibilities, the possibilities will simply grow as better quantum computers come up. The possibilities will simply expand with more innovation in this technology, innovation in this area. But in the current context, quantum computing is still in its very nascent stage. But as innovation like Google Willow keeps coming up, we have other innovations also in the pipeline, like India also have National Quantum Mission, where we are also having an expectation of creating a 50 qubit device by 2030. IBM also is working on a similar technology. They are developing Eagle 100, which is expected to be revealed this year, which will have, which will be right now the fastest quantum computer in the world. That's what they claim. So when all these innovations keeps happening, we'll see more and more possibilities, more and more applications coming up in future related to quantum computing. Now in the context of Google Willow, this much information is enough, and you guys can give this question a try and write your answers. Whatever you think is correct, you write in the comment box, and you can even try writing an answer like this: what are quantum computers? How are they different from traditional computing? That's what I explained. And what potential application does it open up in various industries? Illustrate your answers with suitable examples. This is a question that you can try writing, and when you write it, you can simply email it to me. You can read about my email address, my contact information in the description box. And now let's move to the second innovation. The second innovation is sonobuoy. Now what is this sonobuoy? Sonobuoy is an American undersea domain awareness device. It's nothing but a simple sonar device that is simply going to be dropped from air into the water, and it is expected to give us a full scale analysis, a surveillance of the undersea the undersea location, undersea environment. Now let me quickly talk about what exactly a sonobuoy is, then we'll discuss what is really in the news. So to discuss sonobuoy and its significance, so what is it? It is, you can simply write down, it is an airdropped sonar surveillance device. It is an airdrop sonar surveillance device. So it's a device that you can drop from an aircraft onto any water body, mostly sea, oceans, and it uses the principle of sonar. It uses the principle of sonar, that is sound navigation and ranging, in order to provide a good surveillance map of the sea. Like for example, look at this. So let's suppose, so this is how the sonobuoy looks like. It is nothing but a rod like equipments which are basically dropped on the sea surface. So let's imagine if this is the sea surface and this is where you have dropped sonobuoy, it will eventually throw sound waves. It will eventually throw sound waves into the sea, and as the sound waves are going to hit targets like submarines, if they are going to hit targets like ships or any other deployment of undersea and any other undersea deployment, it will be hitting those deployments, and then it will be reflected back to the sonobuoy. And once the sonobuoy would receive any sound reflecting back, it will study that sound and give you an entire map of the undersea, right? So this is a very innovative technology which gives us a very good command on the overall undersea area surveillance or undersea domain awareness. We call it UDA. So this is the primary application of sonobuoy. It is a surveillance device that once deployed can give you a very good coverage of the undersea environment. Now, what is the reason why sonobuoy is in news? So this is what the sonobuoy is and why it is in news. So guys, there are basically two companies. One is the American company called Ultra Maritime of USA, and we have Bharat Dynamics Limited, BDL of India. So BDL of India and Ultra Maritime of USA. Ultra Maritime has been already manufacturing sonobuoys for American Navy, but now there is going to be a joint manufacturing facility developed where both these companies are going to jointly manufacture sonobuoys, and they will be used for both interchangeably and interoperatively between India and US Navy. So both Indian Navy and the American Navy are going to use the jointly developed sonobuoys. What is a significant advantage here is that once we create such type of machinery, the first significant advantage here is that it is going to support the Make in India initiative of the country. First of all, the Make in India initiative is going to be supported. Secondly, it will help in maintaining higher standards of manufacturing. So because it is going to be jointly used by Indian Navy and the American Navy, by US Navy, so you can expect that they will have to satisfy standards of both the navies. So India will have an opportunity of learning about higher standards of manufacturing. And lastly, if you talk about the importance of Indian Ocean region for us, we already know that Chinese submarines have been sighted in the Sri Lankan waters, and they have been sighted very close to the Indian waters. It is very important that we have a very good undersea domain awareness about the entire Indian Ocean and especially the Indian Ocean region regarding the Chinese incursions in the region. And then secondly, guys, when you talk about the Quad initiative between India, USA, Japan and Australia, when you look at these countries, Japan, Australia, USA, all three of them already have some type of undersea surveillance devices, whereas India still lacks a reliable technology of this nature. So sonobuoy can easily help India in filling up that technological gap. So this would be a good initiative in order to have anti-submarine warfare system developed. It is going to be a better initiative in the context of creating a wide-scale undersea surveillance environment. So this is what we have under the sonobuoy initiative and the sonobuoy joint initiative of development between India and USA. Now, I don't see a very possible question from this particular technology in the prelims either or in the mains, but you can obviously use this as an example while writing answers related to defense technological development of India. You can use them as examples in writing answers related to international relations topics in the mains exam. So you can use them as examples there. Now, the next topic of discussion is microRNA. Now, microRNA, it is an important news of the Nobel Prize on Medicine in the year 2024. The Nobel Prize for Medicine in 2024 went to these two scientists. One is Victor Ambros and the other one is Gary Ruvkun. Both these scientists have been awarded for the discovery and study of microRNA. They have been awarded the Nobel Prize for discovery of microRNA and its role in post-transcriptional gene regulator. Now, what is this post-transcriptional gene regulator? What is the microRNA? Let's talk about it and we'll be able to understand the things easily. See guys, when you talk about the human body or when you talk about any living organism, we all know that every character of a living organism happens to be a protein character. So every character that we have is a protein character. Somebody who is tall, they are likely to have produce more growth or proteins that helps them to grow taller, more growth hormones. Somebody who's short, they happens to create less growth hormones. Somebody who has a good muscular physique, even without going to the gym, they are likely to have those proteins that will help in building muscles, mostly called myoglobin. Somebody who's thin, even they have a good diet but still maintain a very thin physique, they happens to be having a certain gene which produces more myostatin. Myostatin is a protein that reduces the muscle mass of the body, right? So every character that we have is actually a protein character. And we all know that all these proteins are actually secreted. All the proteins that we have in our body, they're actually secreted through the DNA that we have in our body. So every genetic character of our body is actually our DNA configuration. It's basically because of the configuration of our DNA. So every genetic character, it is based on the configuration of our DNA, right? And all this DNA configuration produces different proteins, and that basically creates our external characters, right? So DNA is our primary biomolecule. It is our primary building block. Now, what exactly is the contribution of these two gentlemen? Where is RNA? Where does RNA comes to a picture? Let's try to understand. So guys, what happens here? Our DNA is a double-stranded helical structure. Our DNA is a double-stranded helical structure. Now, in order to produce proteins, in order to produce the proteins that will shape all your characters, the DNA directly does not produce protein. The DNA directly do not code or do not produce proteins. What DNA has to first undergo? The DNA first undergo a step known as transcription. Transcription. The first thing is transcription. What is transcription, guys? Transcription is the process where the DNA turns into RNA. Where the DNA turns into RNA. I'll write it down here, right? Where the DNA turns into RNA. And this process is called transcription, right? So transcription is the process where the DNA turns into RNA. Now, what exactly is RNA? RNA is a single-stranded helical structure. It's a single-stranded helical structure. So DNA is a double helical structure. It basically opens up the two strands, and then each strand secretes a respective RNA, and that process is called transcription. Now, RNA is also nothing but a single-stranded helical structure, right? Now, because the DNA carries all our genetic information, the DNA is having a specific structure. It has a specific arrangement, right? Arrangement. So if I talk about what the DNA is primarily made up of, the DNA is made up of basically three things. There are three things that makes up the DNA. It is called sugar. There is a specific kind of sugar that makes up the DNA. In the case of DNA, we call it deoxyribose sugar. Then there is a phosphate group, or you can just consider phosphorus. And thirdly, it also contains some nitrogenous bases. Nitrogenous bases. Guys, these nitrogenous bases are also having names. There are 4 nitrogenous bases. We call them adenine, thymine, cytosine, and guanine. Adenine, thymine, cytosine, guanine. We also write them as A, T, C, G. These are the 4 nitrogenous bases that are found in your DNA. So basically, how is the DNA structure made? It's a double helical structure like this. It's a double helical structure like this. These strands, they are mostly made up of sugar and phosphorus. They are mostly made up of sugar and phosphorus, right? Sugar and phosphorus are basically found in the strands of the DNA. And how these strands are held together? How are these strands held together? These strands are held with the help of nitrogenous bases. So they are nitrogenous bases holding on to each other. All these strands are held alongside each other with the help of these nitrogenous bases. So guys, these bases are the ones that creates the interlocking of the 2 strands, and the strands are primarily made up of sugar and phosphorus. Guys, in the same way how we have the DNA, we have got RNA as well. RNA is also of the same nature. RNA is also made up of the same 3 things. Even this is also made up of the same sugar. Some sugar might be there in the RNA. Phosphorus is still there in the RNA, right? And it also has got nitrogenous bases. Nitrogenous bases, right? Nitrogenous bases. But the names of these nitrogenous bases is slightly different. RNA also have got adenine in the same way how we have adenine in the case of DNA. Guys, it is not at all required to know what is adenine. Purines, pyrimidines, all these things are not at all required right now. So adenine is one type of nitrogenous base. Mind my words, it is not at all required to know what exactly is adenine, what exactly is thymine, cytosine, guanine. You just have to know that there are some chemicals that holds your DNA together, right? That holds the 2 strands of the DNA together. So adenine we have, then we also have cytosine in this case. We also have guanine here. So adenine, cytosine, guanine are the same nitrogenous bases that we have in the DNA as well. But in place of thymine, RNA have got another chemical called uracil. Uracil is the name of the nitrogenous chemical present in RNA in the place of thymine. So if I would mark them, we normally mark them as AUCG. We normally call them as AUCG. So guys, what happens when you look at the DNA? When you look at the DNA, in our DNA we have got 2 strands of sugar and phosphorus, and both these strands are held together with the help of some chemicals which are nitrogenous in nature. Nitrogenous chemicals, we call them nitrogenous bases, also refer them as ATCG. It is not at all required what exactly is ATCG right now from UPSC point of view, not at all required. So guys, this is what the 3 substances make up or creates our DNA, these 3 things together. Now whatever character do we have, whatever character that makes me different than you, makes you different from the other person, what makes us unique, what makes us exclusive, is mainly because of the arrangement of these nitrogen bases. So all of us have the same sugar and same phosphorus making up the 2 strands, but the way how these nitrogen bases are arranged, the way how these bonds are arranged between the 2 strands, that is not exactly similar for all of us. That is not going to be exactly similar for all of us. All of us are going to have different arrangement of the strands. Hamara strands ka arrangement, the arrangement of the strands is going to be exclusive. It's going to be different for all of us. I hope all of you guys are able to get it. So in one particular DNA, we'll have a different strand, and that would be exclusive for all of us. It is never going to be 100% same. It could be same up to an extent. It could be same up to a very large extent. 99.9% it could be same, but that 0.1% is good enough to create all the differences among us. So this arrangement of the nitrogenous bases is not going to be exactly same. Now, in the process of protein synthesis, when the DNA converts, when the DNA transcribes into RNA, the RNA is going to be a single stranded structure. RNA will also have nitrogenous bases. These bases would simply be here. These nitrogenous bases would simply be here, but they will not be bonding with anything. They would simply be at their own place. They will not be creating any bonds. Like in the case of DNA, they are going to create bonds, and that's how the double helical structure is created. In the case of RNA, is going to be a single helical structure like this, right? That's what simply RNA is. Now what happens once the RNA is created? Once this RNA is created, it is this RNA that eventually produces proteins, and this process is called translation. This process is called translation. So I'll go again step by step. First we have got the DNA. The DNA first produces or synthesizes RNA. This process is called transcription, and then once the RNA is created, then RNA is going to be translating into proteins. It will produce proteins, right? Now there is a lot of other elements or a lot of other chemicals required. There is something called ribosome and all those things. It is not required right now. So DNA converts into RNA, and then RNA converts into proteins. That's it. Transcription and then translation, right? So this is how our proteins are normally synthesized. Now guys, what happens? All the characters that we have in our body, they are going to be produced in this way. So if any one of you has got a certain character that makes them tall, all is that because of the nitrogenous bases in your body. They will first undergo transcription, produces some messenger, produces this RNA. By the way, guys, this RNA is called messenger RNA. mRNA we call it, messenger RNA, small m we write it. And this messenger RNA, so it carries the message of the DNA. So DNA has all the information. The message of the DNA is carried by the RNA, and then this RNA is going to be help in synthesizing. It would be participating in the synthesis of proteins, and that's how the message of the DNA gets converted into the protein. So if your message, the information that your DNA carries, if it is carrying a higher information about height, you will be a taller person, and your RNA will be producing those proteins. If the message of your DNA is carrying high secretion of muscle-based proteins, you'll be a muscular person, right? Even though if you do not go to the gym, likewise, right? So this is a simple way how the DNA first undergoes transcription, then undergoes translation. I believe all of you are able to understand, and it produces certain respective proteins. Now, where does these two gentlemen come in? So where does this microRNA comes up? So guys, when we look at this entire DNA, we have been able to realize that in this DNA, when synthesis of RNA is happening, when this DNA is undergoing transcription into RNA, we have realized that there are creations of some microRNAs also. There are creation of some microRNA as well. What is a microRNA? As the name suggests, a microRNA is a small RNA. It is not a completely big RNA, it's a very small RNA, and these microRNAs that are synthesized, they happen to be created naturally. First of all, they are created naturally, and when these microRNAs are synthesized, these microRNAs, they play a role in obstructing the secretion of proteins. They create a role in obstructing the secretion of proteins. How? Let me tell you. So guys, just visualize that there is a DNA strand. It has cut open itself, produced a respective RNA, and then this is the RNA that is going to produce a certain protein for you. Let's suppose this is the RNA that is going to produce a certain protein. Now, in order to produce a certain protein, this entire RNA is required. The entire RNA is going to be required to produce a certain protein. Now, what happens in the case of microRNA? If there is any creation of microRNA produced, and these microRNA are going to be small RNAs, so these small RNAs that are produced as microRNAs, if they are produced, then before this RNA translates into proteins, these microRNA often go and attach with the RNA. They often go and attach with the RNA. They often go and create a bond with the RNA. And as it creates a bond, the entire combination of the RNA that was supposed to be required to produce proteins, this protein secretion stops. this protein secretion is hindered because of this small microRNA that basically stops the protein from getting secreted. So these microRNAs are often produced in the human body alone. We have already been able to identify more than 1000 microRNA combinations. We have already been able to identify more than 1000 microRNA combinations. So there may be a possibility that these microRNAs can also help in stopping some essential proteins from getting secreted in the body. A person may not be carrying any genetic disease, but because of a presence of a certain microRNA sequence, some essential proteins may not be getting secreted. I hope you are able to understand, because the microRNA hinders the production of proteins. So if I read this particular expression here one more time, these Victor Ambros and Gary Rufkin, they got the Nobel Prize for the discovery of microRNA and its role in post-transcriptional gene regulation. That means once the transcription of the DNA has been completed and there is a production of mRNA, messenger RNA, how this messenger RNA stopped from secreting a certain protein, it is this microRNA that stops them. I hope you are able to understand. So these microRNAs are the ones that are responsible. So I'll try to explain this with the help of pictures. So guys, a human gene on an average contains nearly 20,000. A human DNA, entire DNA of the human body contains nearly 20,000 genes, and these 20,000 genes are going to be, in the form of our DNA. So like I said, the DNA first undergoes transcription, produces a certain mRNA. These are 4 different types of genes, so they will produce 4 different types of mRNA, and all these 4 different types of mRNA, they are going to produce some respective proteins. So they will be translating into proteins. You can see the words transcription, translation here. They are producing into different proteins, and these proteins are going to give shape to different characters. Like one protein may be carrying the characters of your brain cells, another one may be carrying the characters of your skin cells, another one may be carrying the characters of your gut health, intestinal health, and some other things will be carrying the cells of your muscles. Likewise, they will be carrying different types of proteins, and they will be giving shape to these different types of proteins. With the impact of microRNA, these microRNA often, like you can see, the mRNA, when it is supposed to produce a protein, an entire mRNA sequence may be required. But if there is any microRNA secreted in the same DNA strand, and suppose this microRNA, in order to neutralize itself, quickly attaches with the mRNA, then the entire protein sequence or the entire protein secretion is going to be stopped, and this protein could also be an essential protein for the body. And this is how the microRNA helps or works as a gene regulator as well. Some important genes may not be manifested, but you guys can think there are a lot of genes that may also induce some life-threatening diseases as well. Suppose if a person is likely to develop cancer, so this mRNA or microRNA can actually also help in regulating the onset of that cancer by reducing those cancer causing proteins. I hope you are able to understand. So this is something that can effectively cause impact on development of the person. So you may not develop in the same way how everybody else in your family has developed because of presence of certain microRNA. It may help in changing the functioning of your body. Your physiological functions may change, and also it can help in causing and even regulating diseases, especially like cancer, diabetes, the diseases which are normally not caused due to infections, but caused due to a poor lifestyle or unfortunately poor genetics also. I hope you guys can understand. This is what is the role of microRNA, and because of their work in the field of microRNA, they have been given the Nobel Prize for Medicine 2024. Now, what is further more interesting in this study? So microRNA, I believe all of you have been able to understand, but what is even further more interesting in the study is how they have discovered it. So these people have particularly used a kind of a parasitic worm. They have used a type of a parasitic worm. It's a round worm. Round worms, if you consider the biological classification of roundworms, we call them nematodes. Right? Nematodes are basically parasitic worms, and these there are Ascaris, earthworm, sorry, not earthworm, ascaris, roundworms, tapeworm. These are all examples of nematodes. And these nematodes, they have been often used. They are often used in order to do genome studies. Why? Because first of all, their DNA is somewhat smaller. You can easily study the DNA. They only have 6 chromosomes. They only have 6 chromosomes. So the entire chromosomal study can also be done. And on top of that, because of their simplified design, they have all the similar cells like a human body. So we can easily study different DNAs and study the different proteins produced by those DNAs. And by studying them, we can easily replicate the studies in humans, and we use the data acquired from their genomes and to identify and study human genomes, right? So because of the complex nature of their DNA and also simple way of understanding. This is another question that I have been able to put here, and please try to answer this related to the subject of microRNA, and give me your feedback, give me your answers in the comment section. Now guys, next is Twigstats. What is Twigstats? Twigstats is another news that was there in the last week, very popularly covered in almost every newspaper. So what is Twigstats? Twigstat is technically a new tool. It's basically a software, guys. It's nothing but a software, but this software uses high resolution. This software uses high resolution genetic information and tries to find individual level ancestry. It is a very effective tool so far developed to create or to trace individual level ancestry. Guys, it's very obvious. If we have an exclusive DNA, then my DNA has been introduced or my DNA has been produced by my parents. So my DNA is 50% father, 50% mother's DNA, right? Ideally, right? I'm not considering mitochondrial DNA here. So technically my DNA is 50% father's DNA, 50% mother's DNA. Now, if you look at this DNA, and my parents' DNA is 50% of my grandparents' DNA. So likewise, the entire DNA keeps running into a certain family, so if I would try to trace my ancestry, my entire DNA sequences today can help me trace an ancestry back as far as we have the available samples. So when you talk about this ancestral studies, when you talk about studies of ancestry, study of lineage, Twigstats is the latest tool in this particular area. It's the latest tool in this field. And what is this tool special about? So this is a very effective tool in tracing individual level ancestry. It has a much higher accuracy in terms of predicting the right ancestral lineage. It has a very high accuracy in predicting the sequences, the different types of genomes, intermixing, cultural mixing, interracial mixing, all these kind of things. They have been able to predict more accurately, because when you talk about the traditional tools, traditionally we used to identify lineage or ancestry through a technique called single nucleotide polymorphism. Polymorphism is simply mutation, guys. Single nucleotide polymorphism. So when it comes to the studies that often are done for ancestral lineage, we often used to look for single nucleotide polymorphism. What is single nucleotide polymorphism? Basically, it is nothing but mutation or change in a single DNA nucleotide. Single DNA nucleotide. So basically, the DNA is a double helical structure. All of us have understood that DNA has got this double helical structure, and this double helical structure has got the base pairs of nitrogen base pairs are there likewise. So we have a lot of base pairs of nitrogen like this. So what is SNP, single nucleotide polymorphism? Single nucleotide polymorphism simply identifies one single pair. So what is single nucleotide polymorphism? Guys, a nucleotide is nothing but the DNA is also called nucleotide. So if I say sugar plus phosphorus plus nitrogenous base, all these three things together is called a nucleotide. All these three things together is called a nucleotide. So when I talk about single nucleotide, that means a single pair of nitrogenous bases. Sugar would be there in both the strands and phosphorus will be there, and you simply take a link of two nitrogen bases. So a single pair of nitrogenous bases, we call it a single nucleotide. And what is polymorphism, guys? Polymorphism is nothing but change in structure. Morphic is structure only, so it is nothing but a mutation formed in the single nucleotide. So guys, there may be a possibility that like we all are results, we all are hybrids of our parents, 50% father, 50% mother. So if I am 50% father, 50% mother, then what is the case? I will have the DNA taken 50% from my father, 50% from my mother. But there is a possibility that when this DNA is going to create all its entire sequence, there might be a possibility that a single nucleotide might have got mutated. A single nucleotide might have got mutated, and that single nucleotide mutation or polymorphism is good enough to create certain differences, certain different changes in my body compared to my parents, in my protein synthesis compared to my parents. There may be a possibility that my parents might have had a higher metabolism, but I may end up having a slow metabolism because of a single nucleotide polymorphism. So when there is a mutation in a single sequence or a single DNA strand or single DNA nucleotide, in that case it's called SNP. So guys, past studies used to normally study the lineage by studying these SNPs. They would identify SNPs in the artifacts, in the DNA samples or any kind of samples found in the historical monuments found in various sources, and they would try to see how their gene was a little different than our gene, and then they would try to create lineage, that how they are related to the like if I consider Africans, how they are related to the Negreto race, how they are related to the Western Brachycephalus race, how they are related to Nordic race, and likewise, right? So this is how SNP used to work. But now in the case of Twigstats, we don't just follow SNP, we don't just follow the SNP data, but we also look for halophytic data. What is halophytic data? Halophytic or halophytic data is basically the DNA that are shared, right? So they basically take up the DNA of two related, so shared DNAs. So they not just take the data from SNP, but they also take the data of different organisms or different people who have shared DNA. That means they have DNAs of their ancestors, or they are cousins, or they are basically related somehow with each other. So they also take that data, and then they try to combine. Then they try to take up all these things and study what possibilities of lineage or ancestry are there. So with this multiple data sources, they have an improved accuracy in terms of genetic studies. Their accuracy is far more improved compared to all previous studies, and it has been launched by Francis Crick Institute. Francis Crick, guys, Watson and Crick were the two scientists who discovered and studied discovered the shape of the DNA. Francis Crick was one of the scientists, and after his name this Crick Institute has been named. It basically used time stratified ancestry analysis. So in a specific time frame, what kind of DNA, what kind of genomes would have been present in a certain area, and how they would have got intermixed, how they would have got culturally mixed, how they would have migrated from one place to another. All these things can be done with the help of Twigstats. So using this Twigstats, researchers have reconstructed the genetic history of around 1556 ancestral DNA samples, ADNA's ancestral DNA samples from 500 BCE to 1000 AD. 1000 AD. This is considered as the pre-Viking era of the US, of Europeans, of the Scandinavian countries. So they took samples of historical samples, they took samples of current people, and they took a lot of samples, and they also took samples of people who are belong to the same race, their shared DNAs, and that's how they have been able to study. And what they found out, that the Scandinavian countries, they have been able to trace how the population of the Scandinavian countries have moved in Europe, how they have moved into the Baltics, how the Baltics people have moved to the Scandinavian countries, then how the Scandinavians moved towards Britain, moved towards France, Dutch countries, all these areas they have been able to study. And with this, they have also been able to study racial mixing, cultural mixing. And guys, a similar study of this nature also happened in India in the year 2009, where they found out how North Indians show a little more resemblance, a little more, you can say, not, I would not say that they are closeness, but a little more resemblance in their DNA with that of the Central Europeans. And South Indians, although they are very close to North Indians, but they still are having less resemblance and they have an exclusive group in that nature, right? So a similar study of this kind happened in the year 2009 as well. Now, guys, if UPSC would try to ask a question on Twigstats, this term itself is a very popular term which can be often confused by students. So I have given a question on Twigstats, and you guys can give it a try and give me your answer in the comment. I believe you should be able to answer this easily. So yeah, now let's move to the next topic. The next topic here is the hydrogel therapy, injectable hydrogel therapy developed for breast cancer. Now, this is a very interesting study. Why? Because I hope all of you know that since 2011, we are seeing a very high jump in the cases of cancer across the country. In fact, we now identify 4 diseases, non-communicable diseases, that includes diabetes, cancer, hypertension, and cardiovascular disorders. So when you talk about these 4 diseases, cancer is one of the major diseases which constitutes almost 21% of deaths in India. I'm talking about all these non-communicable diseases together. They constitute 21%, and out of these non-communicable diseases, cancer is one of the most lethal disease, right? So when you talk about cancer, we all understand that cancer is basically uncontrolled multiplication of infected cells. So there could be a certain infection in your body. Your immune system likely is supposed to act on it, and it will eventually kill that infected cell and remove it out from the system. But sometime if the infection persists, your immune system eventually may get corrupted and it may stop identifying that cell as an infected cell. And once that happens, the infection starts to multiply, the infection starts to grow, and it can, if it grows, let's suppose in the stomach of the person, the person may be diagnosed with stomach cancer. If that person grows in the lungs of the person, that may be diagnosed as lung cancer. If that grows in the intestine, it will be diagnosed as intestinal cancer or bowel cancer. Or if it grows in the breast, it would be identified as breast cancer, right? Now, normally when you talk about cancer, there are 3 therapies, right? Normally there are 3 therapies for cancer. Whenever you talk about cancer-based therapies, the most common therapy is chemotherapy. Chemotherapy happens to be the most common therapy. In the case of chemotherapy, a doctor would call a patient for an appointment, and they will inject a lot of chemicals into the body. Now these chemicals will go through the blood to rest of the body, and wherever the cancer cells are, wherever the cancer tissue is, the tumor is going to be caught through the blood, and the cancer cells will be killed eventually, right? That's how this chemotherapy works. But you all understand that chemotherapy comes up with a lot of side effects, because the chemicals are going through blood to every organ of the body, every part of your body. If it goes to the healthy cells, the chemicals are still going to impact the cells in the same way, and a lot of healthy cells may get affected. It further degrades the immune system of the person, and you might have heard that in very later ages, if a chemotherapy session is taken, it could be very difficult for old age people to even recover, right? So it's a quite painful therapy. The other therapy is surgery. Now, surgically removing cancer is also a potential therapy, especially when you talk about breast cancer and mouth cancer in India. These are the 2 most common types of cancer, by the way. Breast cancer in females and mouth cancer in males. So if you talk about surgical removal of cancer is a very commonly used therapy for these diseases. But you guys can understand, if there is surgical removal of cancer, you can remove a part of the jaw, you can remove a part of the breast. But what if the cancer relapses and it starts spreading, or if a small amount of cancer would have spread into a nearby organ? Like, you can't take out the lungs, you can't take out the heart. So if it starts affecting other organs also, then in that case, the cancer has started spreading into the rest of the body. And that becomes difficult, because now surgeries cannot help you to remove all the organs of the body. You can't remove all the other organs. You can remove some organs to some extent, but not all the organs to all extent. So that is where comes a very good alternative in the name of hydrogel based therapy. Now, before I talk about this therapy, let me first quickly talk about what exactly is a hydrogel. Hydrogel क्या होता है? So guys, when you talk about hydrogel, hydrogels, as the name suggests, hydro means water and gel means it's a jelly like substance. It's a gel like substance. So basically, what is a hydrogel? It's a polymer, guys. It is a polymer which is hydrophilic in nature. So it's a hydrophilic polymer. What do you understand by the word hydrophilic? Hydrophilic means it attracts water. It is water loving. So it's basically a polymer that can hold a lot of fluids, that can hold a lot of water. That's basically what hydrogel is. So hydrogel is a polymer that can hold a lot of fluids. काफी सारे fluids को hold कर सकता है. It can hold a lot of fluids, right? That's what hydrogel do. Simple, right? Now what the scientists at IIT Guwahati did, although it was a joint study from IIT Guwahati and Bose Institute, Kolkata. Now what these two institutes or the scientists of these institutes did, what they did is they identified the cancer tumor cell of the patient. So they were treating the breast cancer in case of in this case. So they identified the tumor cell, the tumor tissue of the patient here, and then what they did, once they identified the tumor tissue, they injected hydrogel with medicines in it. Remember guys, hydrogel is naturally occurring also. There are a lot of hydrogel that are naturally found. Many of you must have heard of it. There is one hydrogel called gelatin. It's a naturally occurring hydrogel. There is another hydrogel called agar, found in algae, agar. Then there is another hydrogel called chitosan. All these are different types of naturally occurring hydrogels, and hydrogels can be artificially produced also, right? We have polyvinyl alcohol, PVA. Many of you might have heard polyethylene oxide, PEO, right? There are different types of artificially made hydrogels also, but these are the natural ones, and these natural hydrogels are non-toxic, guys. They are non-toxic. They don't cause any dangerous impact on your body. So you can inject them, and because they are able to hold a lot of fluids and you can inject them, your immune system will not get triggered to kill them. They will not be killed. So they can easily be injected in your body. They can easily be put in your body, and your body can easily absorb the nutrients, absorb the water, absorb or take nutrients from the hydrogel whenever required. So what these people did, they basically took gelatin or agar based hydrogel and they mixed the breast cancer medicine into it, and then they injected this medicine into the cancer cells. And here, because it was mixed, targeted to a specific location only, so only to that location this particular hydrogel was kept. Now these hydrogels were basically programmed to start infusing the medicine once they identify a high glutathione levels. What is glutathione? Glutathione is a chemical that is secreted once in cancer cells. So the moment when they would observe high glutathione levels, they will quickly get secreted. They will quickly get secrete the medicines into the tumor cells, and the tumor cells started dying because of the medicines in the hydrogel. Now with this therapy being implemented, it was found that it was much more, far better in terms of side effects compared to chemotherapy, and it is even better in terms of curing a person without distorting their morphology, distorting their structure compared to surgeries. So this has been found to be very, very lucrative, and with this hydrogel based therapy has been popularized. I hope all of you guys are able to understand. This is a simple discussion about this hydrogel based therapy. If you talk about this hydrogel based therapy in India, like I said already, from the Indian perspective, among females, breast cancer is the most common type of cancer, and having more and more alternative therapies available for these cancers, breast cancer, it is even better for a patient, so that they have multiple therapies to take up. There are a lot of other therapies also, guys. You must have heard of CAR T cell therapy. CAR T cell therapy is a popular therapy used for cancer, right? You must have heard of NextCAR therapy, right? There are different types of therapy available for cancer, but everyone does not, become eligible for these therapies. You have to undergo compatibility test. With hydrogel based therapy available, it is supposed to be biocompatible. You may not require any compatibility test for this. Any person can be made eligible, and it is found to be even better compared to other complicated therapies. I hope all of you guys are able to understand. For this also, I have given a question related to hydrogels, and you can try this particular question and give me your answers. The numbers of the questions are mentioned here, so whenever you give an answer, you please specify the number of the question. Now we are reaching to the last topic of our discussion for this week. So here we talk about the ISRO PSLV C60. Now this is the latest mission of ISRO launched. It has been launched by the PSLV, the Polar Satellite Launch Vehicle rocket, and the name of this variant is C60. It is the 60th mission of PSLV. This mission was launched on 30th of December 2024, and this mission is like any other PSLV mission. This is also a very important mission of ISRO, but it is primarily, there are two payloads. Although if you look at all the payloads, there are total 12 payloads. There are total 12 payloads that the PSLV C60 is carrying, out of which 10 of them are from other countries, different organizations, but primarily there are two payloads that are of very high importance from UPSC perspective. One is the POEM-4 and the other one is the visionary SpaDeX experiment. So what are these? POEM-4 itself is having 14 different payloads, and SpaDeX have total 2 microsat payloads. So we'll start with this one by one. First I'll talk a little about PSLV, then I'll talk about these payloads, and then we'll eventually conclude. So guys, when you talk about the rockets of ISRO has got multiple rockets. Most popularly functioning rockets of today's time are three. We have Polar Satellite Launch Vehicle. This is the very popular launch vehicle of ISRO. Then we have got the geosynchronous satellite launch vehicles. This is also quite popular, especially for heavier or much bigger satellites, GSLV. And then we also have SSLV, which is meant particularly for small satellites. So we have PSLV, which is called Polar Satellite Launch Vehicle. We have got GSLV, which is called geosynchronous satellite launch vehicle, and then we have SSLV, which is called small satellite launch vehicle. So these are the three launch vehicles that we normally use. We normally identify these launch vehicles on the basis of their payload capacity. What is payload? Whatever a vehicle carries, it is called its payload. So we identify these three launch vehicles on the basis of their payload capacity. So SSLV, its payload capacity is 500 kgs. That means the total weight that SSLV can carry apart from its own weight is maximum 500 kgs. For GSLV, it can go up to 5000 kgs. There are of course different variants. It can go up to 5000 kgs. And in the case of PSLV, Polar Satellite Launch Vehicle, it varies from 1200 kgs to 1800 kgs. It varies from 1200 to 1800. In some books it may be written 1400 to 1800. Does not make much of a difference here. So PSLV, GSLV and SSLV, these are the three types of launch vehicles that we have, and all these three launch vehicles have been identified based on their payload capacity. Now other than their payload capacity, there are some technical differences also. Like here I'm only going to concentrate about PSLV. When you look at our PSLV, the PSLV rocket, now in general every rocket has a typical design. Every rocket almost has a typical design. In the case of our PSLV, this is a typical design of a rocket. This top end, the top of the rocket, we call this as heat shield. This is called the heat shield. Why heat shield? Because this is going to protect the rocket from the external heat. And inside the heat shield, we put the payload. The payload is kept inside the heat shield. And this rest of the rocket is the actual rocket, guys. This is called the rocket core. Now when you look at the rocket core of PSLV, we have total 4 stages in the rocket core of PSLV. I'm talking only about PSLV, guys. So this is called the PS1 stage. This is the polar stage 1, right? First stage. This is the polar stage 2. This is the polar stage 3. And this is the polar stage 4. Polar stage 4, right? PS4. This is the polar stage 4. So polar stage 4. So likewise we have 4 stages. And guys, the entire rocket is propelled with the help of some nozzles, and sometimes if the payload is very heavy, we may have to put a strap-on motor also. So strap-on motors or strap-on boosters are attached in order to lift an even heavier payload. So we have to put strap-on motors to support a heavy payload. We call them strap-on motors, right? Strap-on motors, okay, to carry a heavier payload. So guys, when it comes to our PSLV, this is how our PSLV normally looks like. So whatever we are carrying, this POEM, this SpaDeX, all these payloads, they're normally put up in this particular, the top shelf, right, in this, inside the heat shield. I believe all of you guys are able to understand up till here. Now, what is so special about the C60? What is so special about C60? So guys, there are a lot of issues that we face when it comes to our space launching. Like when it comes to launching our spacecrafts or launching any rocket, one major challenge that we often face is how to minimize the cost of launch. How to minimize the cost. And second is, can we carry even more payload? Can we increase the payload capacity with the same cost without increasing the size of the rocket? And third, which is very important, is the high pollution that we have caused in the space. Now, in our past videos also, we have very largely talked about space debris. So space debris happens to be another very major problem, right? This is all the kind of junk that have been revolving in space. And guys, I believe you already have gone through the recent articles. There have been a recent article in the Hindu newspaper regarding the Kessler syndrome, which has talked about Kessler syndrome, and this has talked about that how, Don Kessler of NASA warned us way back in the 1970s that if you keep putting a lot of waste in space, a lot of junk in space, this junk will eventually make it very difficult for future projects to be launched, because they would always be at a risk of colliding with this very fast moving junk. So these three challenges are very practical challenges that any country is today facing. One is the high amount of junk. Second is how to increase the payload capacity without increasing the size. And third is how to minimize the cost, how to reduce the cost further, right? So these are the three challenges that we normally have. Now, what makes PSLV C60 interesting here? The first thing what they did, they have launched something called POEM. What is POEM, guys? POEM stands for Polar Orbiter Experimental Module. I'll repeat, Polar Orbiter Experimental Module. Polar Orbiter Experimental Module. What is Polar Orbiter Experimental Module? Guys, as we know that our PSLV contains these 4 stages. So when you look at the PSLV, a lot of components of PSLV also become junk later on. They also become space debris. So what ISRO did, they took this 4th stage. They took the 4th stage of PSLV, and by simply when they took the 4th stage already had the propulsion system that will propel the payload. But alongside with the payload, what they did, they have added one experimental module with it. They've added one experimental module. So what they did, the entire rocket is built in the same way, but alongside the 4th stage, they have put one experimental module. They have put one experimental module here, and this experimental module will carry experimental, experiments of ISRO. Let's say, for example, if ISRO is having an objective of testing, if they have created a new material and they want to test this material in the conditions present in space. So space is having extremely high and low temperature contrast. They have almost zero pressure. They have very less impact of gravity. They have no microorganisms. So if you want to test a material in such a condition, they simply take a small module of that material, put it in the POEM module, and test it all throughout the journey when that particular part of the rocket is evolving in space. And as that part eventually re-enters into the earth, it will eventually be recovered from wherever it falls. So that's how this POEM, Polar Orbiter Experimental Module, functions. So this is a very innovative way in order to make the 4th stage of our PSLV somewhat usable, somewhat worthy, where we can put experimental module, test them. If you want to test a new kind of transmission, transmitter transponders, you can simply put those transponders, let this particular component of the rocket oscillate around Earth, and then eventually as it falls, we'll recover that experiment module. So likewise, we can do all these small modules, we can take it up and put it in the PSLV 4 stage. So we will not be compromising. So the benefit of POEM is that we are not compromising the space inside the heat shield. This space is not being compromised. It is completely using the space around the 4th stage of the rocket. That means you are carrying a higher payload now. You are carrying somewhat more payloads without compromising on the overall payload capacity of PSLV, right? So this basically improves the credit worthiness of our PSLV rockets. POEM. Now when you look at the POEM 4, this is the 4th time, guys. It's not like this is for the first time ISRO has done it. We have done it previously also. We have tested different kinds of batteries, we have tested different kinds of materials. This is the 4th such module, POEM 4, and in this module what we have done, guys, although there are total 14 different payloads that we have put along with POEM, in POEM we have put 14 different small payloads, but what is interesting here is that we have put 2 payloads where one of them, very interestingly, guys, remember CROPS, it's called Compact Research Module for Orbital Plant Studies. For the first time, India have been able to germinate plant seeds in space. We have heard this happening previously by Americans, by Chinese, who have been able to germinate seeds on the moon soil, who have been able to germinate seeds in different places. But for the first time, India has been able to achieve full scale germination of cowpeas. These are cowpea seeds, and these cowpea seeds have germinated in space, right? In space they have been germinated. So a very big achievement in this, right, crop. So remember that India has also joined this club now. Secondly, like I said, debris is a very big problem. Space debris is already a big problem. There are previously a lot of articles written on this. In our POEM 4, we have been able to attach for the first time a robotic arm for capturing the debris. So if in future India comes up with a plan of capturing the debris from the space, we already have the first module sent. It will help us in capturing. So once we trace the debris, we'll capture the debris with the help of this particular robotic arm. So these are the 2 payloads that we have been able to fix at the POEM 4 module. I hope you guys are able to understand. Very interesting payloads, guys. Then after this, SpaDeX. Now what is this SpaDeX? SpaDeX is something which is a primary condition if any country would want to have their own human space flight projects, not particularly human space flight, but their own space stations. If India also aspire to have a space station like International Space Station, like space station like Mir, space station like Tiangong of China, it is important that we perform this particular experiment. Now what is SpaDeX? SpaDeX basically stands for Space Docking Experiment. Simple, guys. Space Docking Experiment. Now what is this Space Docking Experiment? Docking is nothing but, guys, but simply bringing 2 vehicles, 2 vessels, 2 components next to next and aligning them and fixing them, bringing them in contact. Like for example, if you're driving a car, and you must have seen in a lot of movies, they perform stunts where they jump from one car to another once the car is brought into full close contact. So that contact, if you perform that in space, where you can transport material, where you can transport astronauts, and you can fix products from one place to another, we call it space docking, right? So normally this becomes a precondition if you want to have a functioning space station. So a space station is like a space capsule. You have that capsule in space, and when you send astronauts from here, the astronauts spacecraft have to first dock to the space capsule, and then only transportation of astronauts, transportation of goods can happen. So this becomes a very sophisticated experiment or very sophisticated task where you have to simply bring in and dock your spacecraft next to a space station. So India, in its aspiration of becoming in creating the next space station, which is expected by 2035 as per the latest news, we are going to perform this. We have sent this space docking experiment. It was earlier decided that on 9th of January they will be testing it. So today could be the date where it would be tested, but as per the latest news, it may be getting delayed a little more, right? But what is needed to be seen here, if you guys can see, these are the two satellites. These two satellites are microsats, guys. Microsats are satellites which are less than 500 kgs. So they both are microsats. Microsats are those satellites which are less than 500 kgs, right? And this is called SpaDeX A, and this is called SpaDeX 2, SpaDeX B. So spacecraft B, spacecraft A likewise. And what they are doing, if you notice that they have this interesting arrangement or interesting configuration built here. So what will really happen, that both these satellites would eventually be traveling together, and slowly they will be aligning and then coming in contact with each other. Guys, we often use two names here, or two words here. The first word is rendezvous, and the second word is docking. Rendezvous simply means aligning, positioning of one spacecraft next to the other spacecraft. So when you simply align and position two spacecrafts next to each other, we call it rendezvous. So rendezvous is basically when they align and position both of each of them next to each other. And docking is basically when you make the actual physical contact. When you make the actual, so rendezvous is basically aligning, and docking means actual physical contact. So when you actually make a physical contact, we call it docking. So this experiment, including rendezvous and docking, was earlier planned to be performed on 9th of January. Hopefully, if it is performed in the coming days, which is very likely, then India would also be having all the capabilities that it would require if we want to transport any goods or any equipment from one space station or one spacecraft to another spacecraft. That's basically called docking and rendezvous. So this is what India was planning to do on 9th of January 2025. And you can understand, if this experiment successfully takes place in this coming week, then I will be surely taking up this topic next week again, right? So again, on this topic, there is one question that I have given. You guys can give it a try, read it and tell me about this answer in the comment section, right? So this was the last topic that I had to discuss with you guys, and this is my personal request to you, that you write answers, and you can write them directly to me. My email IDs are there in the description, so I will be checking the answers myself, and these regarding these questions here, I'll be sharing the answers soon, in the chat itself, in the comment section itself, right? So this is everything, guys, and this is how we are going to discuss science next week again. And next week we'll take up all the current affairs and all the development that happened in the subject of science from the date 9th of January. And I'll see you guys next Thursday now. Thank you so much.